All right, it's time for the anti-penultimate lecture for the Bioinformatics series. We're on lecture number 6, if I'm not mistaken. So today we will be talking about quantitation. We'll be talking about the set of technologies that most frequently show up in biomarker hunts when we're looking at the world of proteins, and when we're looking at the world of metabolites as well. We've talked about biomarker hunts before when we were dealing with data from, say, microarrays and genetic sequencing. But now we're looking at the recognition of metabolites and proteins, and that's most frequently, close our doors here, that is this metabolite and protein world tends to be something that we measure through liquid chromatography, mass spectrometry. So if you'll stay with me, we will discuss this field in that light. Now, I wanted to mention that in preparing my slides in discussing the XCMS workflow, I really benefited from having slides that were provided by Paul Benton and Gary Suziak. This support came, I don't know, a couple of years ago when I was writing a workshop for a corporation in Brazil, of all things. So they've really helped me out here. So we will start with a discussion of the fact that our quantitative data are not guaranteed to be continuously distributed. They may be discrete. They may be numbers instead, count data. So we will talk about some of the challenges associated with that. We will talk about aligning and grouping signals for a particular ion. This is a challenge that comes into effect when we have various chemical forms that may be observed for a particular metabolite. We will talk about the use of spectral libraries for identification. I already mentioned that they have a fair bit of application in the space of proteomics. Today we'll discuss it in the context of metabolomics. And we'll also talk about lipidomics. Those poor guys in lipidomics, they kind of are caught between two worlds, in effect. The proteomics people tend to ignore lipids because they don't have sequences, and the people in the metabolomics world tend to dislike lipids because they're awfully big, and they have a lot of structural diversity. So lipidomics, in some ways, is the fourth omics. Alongside genomics, transcriptomics, and proteomics and metabolomics, the fifth omics. The lipidomics people tend to be even a little separate from the rest of the metabolomics community. So we will talk about their interesting plight as well. We remember the far too confusing diagram that I've made to show the mapping of protein structure and protein identification. We're going to step out of that space, back over into this green space a little bit today. But we will also spend a fair amount of time talking about the problems of metabolomics, being able to deal with NMR and electron impact tandem mass spectra, I should say, and ordinary old tandem mass spectra, as we've discussed before. How do we make sense of the data from these? And along that line, we want to talk about chromatographic time alignment and peak integration. And I think I will have very little to say about flux analysis, but it's important to note that it exists for people who want to model how systems are moving from are transmuting from one metabolite to the next in a long chain. So the people who do mathematical modeling of that sort are involved in flux analysis. So I'm going to introduce the topic of spectral counting. Spectral counting is a semi-quantitative method that we largely use to determine which proteins are changing between a couple cohorts. As I think you remember from yesterday morning's practical, the evidence that we see for a particular protein will usually span multiple peptides that map to that protein. However, it is possible to identify individual peptides in multiple forms. You may identify them in both a +2 charged form and a +3 charged form. You may identify them in an unmodified state, or in a state where a methionine has been oxidized, for example, or carries some other modification. So this means that there's something of a cloud of possible peptides that might be observed for any protein. There are those that show up with very, very high consistency. 9 times out of 10, when you analyze this protein, you will see this set of peptides. But there are other peptides that you might also see that are present at slightly lower probabilities, that you might identify 50% of the time a protein is present, or 10% of the time that a protein is present. proteomics has a lot of chance associated with it. And the instrument is going to select from the available ions that are present right now from liquid chromatography based on whether it has fragmented them already and based on how intense they are. So there's something of a random chance in any particular peptide, whether it does or does not get identified by the mass spectrometer. Think of what happens when you have a sample of high complexity, like a piece of meat out of somebody's colon or arm or whatever, right? When you have a sample of that high a complexity, the number of possible peptides that could potentially be identified is astronomical. And you may run the sample once, twice, 10 times, 20 times, and still be seeing new peptides that you didn't see the first time you ran them, or the second time, or the third time. So the combination of which peptides get identified in any one run is a competitive process. The sample contains far more peptides than the instrument can plausibly identify in any single run, or any 10 runs, for that matter. So this creates a competition in which the peptides that are most intense have the best chance of being subjected to tandem mass spectrometry and subsequently being identified to that particular protein. Remember that both of those are probabilities. So you have the probability that a tandem mass spectrum is acquired for this peptide, and you have the probability that this particular tandem mass spectrum is actually successfully matched to a peptide. Both of those are catch-as-catch-can things. So when we think about this process, the trypsin digestion can differ in which peptides get produced. Which peptides are able to attract protons to them, as I described this morning, can also be a random process. And so in effect, any proteomic sample is going to have variability in which peptides get identified. So the magical bit about spectral counting, then, is that the number of spectra that get attributed to a particular protein in a sample has a lot to do with how much of the total protein content of that sample that individual protein accounts for. So if you have samples of comparable complexity, and that is a requirement, if you see a very different number of spectra accounting for a particular protein between cohorts, you can use that to say, this protein is differential between these two cohorts. If those cohorts are of different protein complexity, maybe this one's 200 or so proteins, and this one's 1,000 or so proteins, you can't make such a comparison anymore, because the competition for the instrument's attention is not as fierce in a sample with 200 proteins as it is in a sample with 1,000 proteins. So we start with the fact that a more concentrated protein produces more peptides through the digestion process successfully, that more of those peptides successfully get protons stuck on them in electrospray. The tandem mass spectra are more likely to be acquired for that protein, and the spectra that are acquired for that protein are more likely to be identified successfully. All of these so a protein that is present at a higher concentration is going to account for disproportionately more spectra that get successfully identified. So spectral counting is not a great method if you're... Did we just go yellow? That was very strange. Maybe the projector's flaking out on us still. I'm going to say only good things about this projector for now. So if you are conducting many proteomics experiments, the number of spectra seen for a protein is likely to be larger when there is more of that protein present in one cohort than in another. So spectral counting is one of the methods that we used, that we compared in a study from the Bioinformatics research group of ABRF a few years back. And one of the things that we discovered was that spectral counting is a method that's relatively hard to screw up. That the people who use spectral counting tended to do better in deciding which proteins are differential between cohorts because the software is not complex. You just have this table, essentially. So we're going to look at some examples of what those look like. So here we are looking at data from the ABRF 2009 study. This is the case where we had red E. coli and yellow E. coli. Did I see a question forming in some minds? I think I did. We can pause. No, we didn't have a question. Well, I'm glad we stopped for it anyway. So, a moment where you get to hold your warm cup of tea and contemplate is not a bad moment in a day's work. So, let us imagine a table like this. Now we have a massive table. We have how many proteins we identified in those E. coli guts yesterday morning? 600, 700, something like that? Large number. So I want you to imagine that this table does not stop at row 15, whatever it is. I don't know how many rows that is. Let's imagine that the number of rows this table shoots down comes to 700 or so, way down here. Now each of these columns represents a separate experiment. These were actually paired experiments in that we did the red one, and then we did the yellow one, and then we did the red 2, and then the yellow 2. Doesn't much matter in this case, but I'm just saying. So what we have in R1 is the number of spectra attributed to protein PO2931. That has a friendly name, but that's the number I have accessible at the moment. How many spectra from this experiment were matched to this particular protein? That's what each cell of this spectral count table represents. So we had 5 different red replicates, and we had 5 different yellow replicates. And as you see, the number of spectra seen for a given protein sometimes differs. But let's just start by looking at just the reds, okay? Just the reds for a moment. Do you see differences in how many spectra were observed for a protein in the red cohort? Okay, give me an example of a protein row that has differences.
PO-A6M8.
Okay, row 3 then.
Yeah.
PO-A6M8. So what is the smallest number of spectra observed for that protein in the red?
45.
45, and the highest?
60.
60, okay. So one of the things that you should be aware of is that there is variability in these measurements. And anytime we're going to find a difference, we have to be able to overcome that variability by the difference between the means, by t-test rules anyway. That's close enough. So if we see a difference like from 45 to 60 on the red side, we would need to see a pretty big difference in the means over in the yellow, from yellow to red, to be able to spot that difference, because the variability is already pretty big. In this case, the variability over on the yellow side is not tiny. It runs from 52 at a minimum up to 60 as a maximum. Those two distributions, red versus yellow, they overlap pretty heavily. And in this case, we would say, any statistical test worth its salt is going to decide, well, there must not be any difference in this protein between these two cohorts. Okay. So having a sensible way to do our statistics on count data really matters. All right. I'm going to ask you a tricky question. I have a protein for which I have very little evidence, but I have some. So I'm trying to do a t-test of 2 spectra seen in each of the red runs, exactly 2 in each of the red runs, and exactly 3 in each of the yellow runs. What's going to plague me if I try to do a t-test of that? Yeah, you maybe it's okay. If you have no idea, it's okay. Yes, I'm asking you. I have a bunch of 2s on the red side and I have a bunch of 3s over on the yellow side. Is that cool? Can I just t-test that and run away with it?
No.
No, she's right. You cannot. Do you have an answer why?
But what do you mean by 2s and 3s?
Imagine that I have a protein that is 2, 2, 2, 2, 2, and then in yellow, 3, 3, 3, 3, 3. Can you just t-test 5 2s against 5 3s?
No.
She's correct. You cannot just run a t-test on that. But why? There are 2 violations of the t-test assumptions that are at play. You're doing really well so far. If you got nothing, just say it.
I don't know.
She doesn't know. Does anyone have R installed on a laptop?
Yeah.
Okay. Well, somebody do a t-test of 5 2s against 5 3s. I'm curious what the result is. And Gerald, I'm going to poke at you next. You're just sitting next to her, so I need to ask you. Why would doing a t-test of 5 2s against 5 3s be a bad idea? Some of it related to the bullet point in the overview slide, slide number 2.
Yes. Yes.
I'm not sure at this moment.
Okay.
Because of there's low homogeneity of variance between the two samples.
That's really good. Well, what is the variance within red and within yellow?
0.
0. Right. Yes. There is no variance. 0. Those are correct answers. So if you cannot estimate variance correctly, you've got a real problem. And spectral counts, when the numbers match across the board, there's no minimum on the variance that you get from that. The variance of 2, 2, 2, 2, 2 is 0. So any difference in the means seems significant if you have 0 variance. It's a big problem. Okay. So 0 variance is the problem I'm looking for, but there are other couple things that show up here too. One is that we need to have a normal distribution of errors. And when you have no variance, you have no errors to work with. So your ability to estimate the variance is a real mess at that point. But also consider that t-test is not created for counts. In fact, I would like everyone to say this with me if you would. Because sooner or later you're going to be tempted, and I want you to be strong against this moment. So together we're going to say, t-test is not intended for counts. T-test is not intended for counts. Great, I feel good about that. There are ways to get around this, but I just would like you to think whenever you start to run a t-test, you take a look at your data and figure out if they're appropriate for that. So we can beat the 0 variance problem. We gave an example of that 2 days ago when we looked at microarrays and we talked about E-Bayes, the empirical Bayes t-test. If you remember Lemma's version of t-test, that an empirical Bayes moderated t-test, we can estimate the variability by looking across all of the proteins, not just this one protein. So that's one way that we can step around the 0 variance problem and not erroneously claim that 2, 2, 2, 2, 2 versus 3, 3, 3, 3, 3 is significant. The next is that t-test and as I mentioned, we don't use it in counts because the t-test is designed for continuously distributed variables, not discrete variables, and discrete variables include counts. Okay, so that said, are we stuck? Are we just really out of luck? And the answer is, well, no. There are some tricks that we can use. Fisher's exact test is one of the examples for this. A couple months ago, I did one of Dave's useful hours on the subject of the Fisher exact test. The mathematician R.A. Fisher was at a party where he encountered a woman who said she could tell whether milk had been added to her tea before or after the tea was. Right? Did she put a little bit of milk in the cup and then pour the tea in and then mix it? Or did she put her tea in the cup and then put a little bit of milk in and mix it? She claimed she could tell the difference just by tasting it. Well, R.A. Fisher thought that would be a really neat trick to pull off, but he was skeptical. He was skeptical that she could tell this difference. The Fisher's exact test came from this little dinner party. So he proposed that, to match our example here, the woman would be given 10 different cups of tea. And in 5 of them, the milk would be added first, and in 5 of them, the tea would be added first. She would know in advance that it was a 5 and 5 mix. Her only job was to take a sip. I can't drink theatrically, I'm sorry. To take a sip of each cup and then say, was the milk first or was the tea first? She couldn't say, I don't know. She had to say one or the other. So the Fisher's exact test is a system built around what's called a contingency table to get a probability that somebody would do this well or better by random chance alone in such a trial. So a contingency table shows up in a lot of places. So the way that this works out is kind of a 2x2 table. And in this case, the lady says milk is first. In this case, the lady says the tea was first. In this case, reality says milk was added first. And in this case, reality says that the tea was added first. So we then fill in that table. There's going to be a total of 10 trials, and so you'll have numbers distributed across those 4 values. From that contingency table, Fisher's exact test will give you a probability that somebody would do this well or better by random chance. All right, so that is Fisher's exact test. The problem is that Fisher's exact test does not take replicates into account per se. The replicates are just counted as numbers here. So if you've got a 5-on-5 table, it boils down to one contingency table, and then you lose the variability information. So that's a little problematic. But Fisher's exact test is useful, especially, now this is kind of a dirty secret, right? Occasionally, in your career, you will be called upon to do an experiment, like a pilot experiment, where you don't even have replicates. You just have one-on-one for a particular comparison, right? You might think, well, I can't run a t-test on this, therefore I can't figure out whether the pilot was worth anything. But Fisher's exact test doesn't care if you've got replicates or not, because it's going to be built off of count data. So keep it in the back of your head for that moment when somebody asks you to run an experiment with insufficient numbers of replicates. You might save your bacon on that score. Okay, on we go. The Poisson modeling. Statisticians have solutions for everything. And one of the useful things that they've done is to set up linear models that are paired with Poisson link functions. In effect, these Poisson link functions allow us to treat count data as continuous distributions after we do this fit with these. So you will see that people have used both Poisson and Fisher's exact methods in doing these kinds of statistics, and they have been very, very useful for this. But in general, what I would say about spectral count-based methods is that people use them to determine that protein content differs, to say this protein is or is not different. It's much rarer that you see people using spectral... I think I've got a loose wire back here. There we go. Well, don't know that I can actually screw this in. Try to fit it that way. Okay. So in general, what's that?
No, just having an OCD moment.
Oh, the flickering yellow? Yeah. Is it going to keep doing it, you think? Or
No.
we'll see. So if you have only spectral count data, you should probably say, I can tell which proteins are differing, but I can't have a very accurate estimate of how much they're changing. There are other methods that have been created for that purpose, though. And probably the big one that I see used all the time in South Africa in particular, and in Europe, is called MaxQuant. It was created by some researchers up in Germany. Jürgen Cox is the lead author on this paper. Matthias Mann, his mentor, was the senior author on it. I should also point out that Jürgen Cox's mentor and my mentor have kind of a long-standing beef in this field. People really want to be remembered as the father of proteomics, and anybody who has a claim to that name automatically hates everyone else who also has a claim to that. So this is one of these odd spaces where I'm talking about the opposition's work. Ooh. So here we are looking at MS intensity as quantitative data. Imagine that instead of thinking only about the counts of spectra accumulated for each protein, we are instead thinking about the amount of intensity that was observed for the peptides of that protein. And that's what MaxQuant is intended to do. Now I'm showing over here at the left real data. These give you an example of the resolution that's possible with a Fourier transformed mass spectrometer, particularly with an Orbitrap. We have 2 of those upstairs over in the clinical building on the 6th floor. So we are looking at a very narrow region here. This is from 468.0 to 470.5. So a total of 2.5 m over z. But look how narrow these little buggers are, right? These peaks are really, really well resolved from each other. This is what you get through Fourier transform mass spectrometry. Extraordinarily high resolution, very, very good mass accuracy, And we have the ability to tell apart these ions. So this is 468.31, 469.31, and 470.29. So what is the relationship between these three peaks, do you think? Anyone notice any defined mass difference there? What is the mass difference between the first peak and the second peak? 1, which suggests a chemical relationship between those two ions. What do you think it is? What would cause two chemical compounds to differ by 1 atomic mass unit? What has the mass of 1? What?
Hydrogen.
Well, it could be a hydrogen, but it's not. It's close. What happens if you merge a proton and an electron? No subatomic physics people here. Okay, that's fine. But what would cause a chemical compound to be 1 mass unit away from another compound?
They're isotopes.
Ah, on the nose, yes. Isotopes. So, about 1 in 100 carbon nuclei is heavy. Is a C13 instead of a 12 Dalton unit. So what we're looking at is the ions of this peptide that are all C12, the monoisotope, in other words. And these are the ones that have 1, perhaps carbon 13 in them. And maybe some of them have either 2 carbon 13s or they have 1 heavy oxygen on board. Probably the 2 carbon 13s is a more likely scenario. So this is 1 chemical compound with the same chemical formula, but a difference in how many neutrons are in the nuclei. Okay, so even so small a difference as that is easily discernible through Fourier transform mass spectrometer, Fourier transform mass spectrometry. If these were doubly charged, how far apart would these peaks be on the m/z axis? 0.5, exactly. So one of the ways in which we discern what the charge state of a peptide is by how far apart its isotopes are. All right. Now, I want you to think about this sort of turned forward like this. Now we've got intensity poking out towards you, and we're showing time as the other axis. So here's a particular peptide ion that came off from that time in this time frame. It was the signal for an individual peak was all between 917.98 and 918.02. So a very narrow peak, and we see this peak has some width. So at the very most intense, where it pokes out the most, it's got the broadest width, and we can estimate what the mass of this ion is variably. We can go at each retention time slice, and at each slice of time, we get a different estimate of what this precursor, what this peptide ion mass is. So viewed another way, we can make a 3D histogram out of this. We have a peak that shows up, abruptly crests, and then fades away. I think I've mentioned that pattern. That's our chromatogram. But here we're seeing that this peak has a bit of width as well, because we don't get just a single spike in the MS scan. We actually have a peak that has a certain amount of width to it. So here they're zooming that out a fair bit. So in effect, instead of thinking of this as this spectrum as either a 1 or a 0, it does or it does not contribute towards a particular protein. Now we have a scalar coming out of it. We have an intensity that is estimated by looking at the volume of this peak. Okay? The chromatogram can be integrated to give us the volume or the area of the peak to contribute towards our measurement of the protein. Now, this chromatogram that I've just shown you represents a rather different way to look at these data. That instead of counting spectra contributing to a protein, we can look at intensities contributing to a protein. But I really want to move us towards the right experiment for the right goal. if you're trying to make a quantitative claim about proteins, you should do a quantitative experiment built around that set of proteins. That brings us to targeted proteomics, one of the key concepts that I want you to carry away from today. So why do we care about targeted proteomics? Why not just do everything shotgun, 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 and just identify lots of peptides and then say we know something? Interesting proteins are low in concentration. Generally speaking, we don't really care about the concentration of albumin changing. From a proteomic perspective, albumin is just always there. It's not the interesting protein. We want those things that are maybe the 1,000th most popular protein in a sample, or those that are at least a few hundred down. The stuff at the top we know really, really well from classical biochemistry by this time. So shotgun methods, these discovery proteomics methods, detect any given peptide with low probability. And instead, we want to have as solid a measurement as we can get for every peptide that we're going to try to measure. So targeting an individual peptide or a set of peptides, we talked about 150 peptides this morning, achieves higher sensitivity and generates a profile of intensity. This is the chromatogram, again, showing up. We have a pattern of intensity in response to time. Right now, if you do a really, really big proteomics shotgun experiment and try to publish it, you will very frequently find a journal saying, well, we don't want to publish that because we need orthogonal verification that these differences you found are real. They want to see you pull together a different cohort of samples, remeasure these proteins that you claimed were differential, and prove it. Prove that they're differential through quantitative proteomics. So lots of people now understand discovery proteomics is great, but they're learning through the publication process that they really need to have a subsequent quantitative experiment to publish. Right now, I'm working with the University of the Western Cape to put together an instrument. We're trying to buy some new instruments over there. So one of the instruments they're buying is a shotgun instrument, an ion trap. An ion trap is a great instrument when you just want to say what proteins are here. But part of that same purchase we're trying to negotiate is the purchase of a triple quadrupole because we want them to be able to do follow-on quantitation to be able to publish their work. This really matters. Now, I've mentioned triple quads a few times. I've mentioned a few kinds of instruments to you. Ion traps and QTOFs and Fourier transform instruments, and now the triple quad. But I'm going to emphasize the triple quad in part because this instrument design has been so stable and so important for the last couple decades. A triple quad is called that because it has 3 mass analyzers. It has, well, as all mass spectrometers do, it has an ion source. This is the electrospray ionization up front. And it has a detector. We talked about electron multipliers this morning. So these are the ion source and the detector. The middle part shows how we can analyze the peptide ions. So up the front, we have Q1. This is quadrupole 1. It's a very inventive name, I know. This is designed to isolate out only to filter out everything but the ions of a particular mass to charge value. A particular mass to charge value. This is defined by the set of transitions you give it. So if you have 150 different peptides that you're monitoring, this first Q1 is going to be cycling through those 150 different mass to charge values corresponding to the 150 peptides you want to measure. Next, you have a collision cell. This is how we break apart these peptide ions to produce fragments. And then only one of these fragments is going to be allowed to pass. Only one m over z value of fragment will be allowed to pass. So in effect, Q1 is screening out anything but our peptide of interest. Q3 is filtering out anything but the transition of interest. This morning I said we wanted to keep track of 50 proteins, 3 peptides for each, 3 transitions, 3 fragment ions for each peptide. So this first Q1 is going to be rotating through 150 different peptide m over z values. Within each peptide m over z value, we're going to rotate through 3 different transitions, 3 different fragment ions that we expect for that peptide. In effect, we get 450 different transitions measured as chromatograms. So in a larger framework, we start with proteins that we've digested to peptides. We've separated those peptides by liquid chromatography. We've produced a electrospray ionization cloud of these ions. This is all very much the same as in the discovery shotgun experiment. But now we screen out all but the target mass of a peptide, blow it apart, screen out all but the fragment mass, and that gives us a trace, a chromatogram, the intensity as a function of time here. We can integrate these peak areas to make them comparable, to evaluate how the intensity of signal differs between two things, and that gives us our judgments about whether this protein is changing in concentration, and to what extent the my goodness, that is really going to get me. I'm going to try swinging this around this way. Okay. Now we are looking at real-world data. I've talked enough about chromatograms without actually showing you one. Have you noticed? Well, here's one. Here we are looking at a range of retention time. This is standard liquid chromatography. We are looking at a fairly narrow region, about 49 to 56 minutes into this experiment. So we're zooming in on one small part of the experiment. We are measuring 1, 2, 3, 4, 5 different fragments or transitions for one peptide. You can see that up in the legend. We've got these 5 different lines that we're going to follow. So if you were only paying attention to the blue trace here, you might say to yourself, my goodness, look at that massive trace. Certainly this peptide comes off at about 50.1 minutes. You'd feel really great about that. But wait, when you remember that this peptide comes off the column at some characteristic time, and that is the time at which all of the chromatic traces, all of these chromatography peaks should rise and fall together. So the fact that we're monitoring multiple fragments for this peptide helps us to realize that this, even though it is not the most intense signal for the dark blue signal, is the correct one, because this is where all 5 of those traces rise and fall in concert. So this is the value for us in measuring multiple fragments for an individual peptide precursor. Okay, so that's what these things look like for real. Real samples have lots of peptides, some of which coincidentally happen to be at the same precursor mass to charge and fragment mass to charge. So being able to look at multiple fragments helps us to avoid making a mistake in finding where that chromatographic peak for that particular peptide really is. Any questions so far? Do we have a triple quadrupole mass spectrometer at Stellenbosch, Tygerberg? Anyone ever been over to the proteomics lab? It's on the 6th floor of the clinical building. Marne Flock is a very interesting guy. He had a lot of interesting things to say. But in fact, the proteomics laboratory at Stellenbosch, at Tygerberg, does not have any triple quads. Not yet. Clinical pharmacology at Tygerberg, however, does have 2 different triple quadrupole instruments. So that's great. And there's a mass spectrometry facility over on main campus that's got a pile of triple quads. So we have the availability of these instruments. If you have particular proteins and you want to know are they or are they not changing in this cohort of, in these cohorts of experiments, you can do this experiment today through CAF, the Central Analytical Facilities. You can also do shotgun. We've got plenty of Orbitrap instruments for you to run over there. So these experimental methodologies are available to you as part of the graduate research that you would hopefully do here. All right, on we go. So I believe you all met Brendan McLean this morning on our fun video of tutorial. I'm not quite sure who that other voice. It sounded like he had dubbed his own voice on top of his own for the video. But everyone makes mistakes when they're making the first training video. So here we are looking at a screenshot of Skyline. I really want you to be aware of this software because if you are ever going to do a quantitative proteomics experiment, it's quite likely you will want to design it in this software. It's quite likely that you will want to analyze it in this software. So skyline.ms is an easy resource to hit. All right. There are quite a few kinds of plots that show up here. And some of these show up, some of these are quite useful in understanding lots of technologies, and I would like you to understand them. So on the left, I have a calibration curve. A calibration curve is quite essential anytime you're working with a quantitative technology. And the reason for this is that you would like to know the range of linearity for that quantitation method. Which is to say, if you put in 1 unit of sample, you would like to know that the instrument is responding with 1 unit of intensity in response, or 1 unit of quantity in response. If you put in 2 units, you would like it to double to say there are now 2 units present. If you put in 3, you want it to put in 3. But if you suddenly throw in 1,000 rather than 3, your ability to determine that 10,000 is 3,000 times the size of 3 is putting quite a strain on most of these instruments for their dynamic range. So there's some range in which that instrument is able to detect that you've given it an analyte, and there's a different range in which it's able to quantify that. So when we look at this plot, on this axis we're giving the logarithm of the peptide concentration. On the other, we're giving the log peak area ratio. So that is its quantitation that's coming back in response to what we give the instrument. At very, very low peptide concentrations, here at 0.0044 nanograms per milliliter, tiny amount, the instrument is actually not able to quantify with any reality. It is able to say there is some amount of analyte here, so we're within the limit of detection, but we are not on the limit of quantitation, because we see that that is only when you hit this diagonal line. So we have to get up to about, let me see, that's going to be about, say, 0.9 nanograms per milliliter before we start hitting that line. And then we see that increases in the amount we shove into the instrument are met with increasing reports of how much there is. This is the need that we have to do calibration curves to understand whether the instrument is accurately measuring what we're giving it. Okay. We would see a similar result, by the way, if we came up here to like 1,000 nanograms per mil, because we would probably run past the instrument's ability to say, to respond more by giving it more sample. So there's also there's a sparsity issue. You're not giving the instrument enough to quantify, and there's also the ceiling issue, right? It's like turning your speakers up on your radio all the way to 11. When you hit that top, you don't really have much, you have a lot of distortion in the sound, and that's because it's not able to raise its volume in a way like that. So you hit the ceiling on what that instrument can detect, and suddenly it flattens out. You're giving it more sample, but the signal's not going up in response. Okay, so that was the calibration curve. Over here on the right, we have a retention time variation. This is something very important to understand. Frequently, the chromatography we use in biology is not really optimized for perfect separation. It's optimized for reproducibility. It's geared toward the ability to be used in a wide variety of analytes. So frequently we find that there's a degree of variability that we see in when a particular analyte comes off of a particular column. Maybe you run that sample 10 times and you see, a few seconds variation each time on the retention times. So being able to compare those across replicates is actually quite valuable. When we talk about mass spectrometry as a quantitative technology, we always have to be a little bit careful. So masters of the x-axis is a statement I picked up off of Dr. Senko several years ago at an ASMS meeting. He argued that mass spectrometry is really, really good at picking out the mass, or the mass to charge value of an ion very, very accurately. But the intensities that we observe, they bounce around a fair bit. So one of the problems that we encounter is the coefficient of variation that we observe for an intensity measurement. If you just shoot the same sample many times over, the intensity that you get back isn't stable. It bounces around a little bit. So in terms of t-testing, you should think in your mind, variation. That is going to be a challenge when I want to tell means apart. And you're right. So we have to keep in mind that the variation of our measurements is going to be limiting on how big a difference must be before we can see it. All right, this brings us to the fine world of metabolomics. We are sitting at slide 16 and 41. I've given myself a fair bit to talk about in metabolomics because this is the only lecture where I really touch this topic. We've talked about shotgun experiments as a discovery for proteomic inventories, and we talk about targeted experiments as a way to quantify in a directed way. This same division between targeted and untargeted metabolomics exists. You may have an experiment where you're trying to use SRM, just the same for metabolites as you would use it for proteomes or for peptides. And in the same way, you might use untargeted metabolomics in order to find ions that differ so that you can characterize them. Now, I know that that's a little different than what I've been talking about with proteins. With proteins, I've generally been talking about producing an inventory of all of these different samples and then asking which proteins in this set change most significantly. That's a little backward from the way we see it in many other disciplines. In their world, they tend to ask which intensities differentiate metabolites. Let's now go identify those differences, those changes. Okay, so it's a little bit back to front there. There are some really good papers out there on this subject. I definitely recommend that you would read some of them if you decide to get into the fine world of metabolomics. Now, some of this information comes from the metabolomics workshop that I did a few years back. And this is kind of the workflow that I envision for an untargeted metabolomic study. We start with peak picking. This is something that I emphasized a lot because this was what my area of research covered. How do we do the signal processing to get very stable values out of our mass spectrometry data? Feature recognition, being able to recognize chromatograms out of these data. And remember, the chromatograms are not measured continuously. We have some amount of time that separates the time slices at which we measure them. If that time slice is 10 seconds rather than 5 seconds, our ability to get accurate peak shapes is going to be curtailed. So, retention time alignment, being able to recognize and account for these variations in retention time across different chromatographic columns, or different uses of the same chromatographic column. Differentiation, to find which peaks actually change in intensity between the two. And then identifying those biomarkers. So only at this last step in metabolomics do we frequently determine what the identities of these molecules that differ would be. XCMS is one of the most widely used data analysis pipelines out there. This morning I was going to demonstrate the R-based run-on-your-own desktop variant of this. It's very, very powerful, and you can do a great many things with it. But most users, I think, are probably a lot happier using the web interface for the software. That's the XCMS Online Scripps EDU. Scripps EDU is a research institute in La Jolla, California. That's where I finished off my PhD, actually, between 2000 and 2003. Lovely place to live. You know, having surfers go by your workplace is pretty cool. Seeing Tiger Woods in your backyard at the golf course right next door was also kind of neat. San Diego's a strange place. Anyway, so you can see that the usage of this system is just growing by leaps and bounds. Gary Susiak's lab has been one of the most substantial in this space of metabolomics for a long time. So one of the first things I was going to demonstrate in the software this morning was the detection of a region of interest. We are looking at differences in retention time. This is how many seconds it's been since we started this experiment. On the y-axis, we have the intensity that's been measured. So if you imagine that the instrument is sampling at more than a hertz, here we've got multiple measurements per second, we're getting different measurements of intensity out of this. And as a result, the chromatogram that comes out is beautifully smooth. That's a lovely looking chromatogram. If you look at this on another axis, though, up the top, you can see that we're measuring, that our measurements have some amount of drift back and forth on m over z value. This is the mass to charge ratio that the instrument is reporting for this ion. So you can see that it's reasonably tight when we get into the middle of the peak at the most intense signal. But as the signal tails off, as we have less and less of it present, the mass is more error prone. And this is something that we see. The more intense the signal, the more accurately you can know its mass in effect. Okay, so defining a region of interest is effectively drawing that box around it, saying these are the parts where where a signal at this mass to charge value is most reliable. We already talked about an example in an earlier figure where we may see different traces for a single chemical compound. That difference came about when we had extra neutrons in some of the nuclei of those molecules. So we were able to see isotopic variance of the same ion, which means that when you're looking at a region of interest, pretty much any metabolite you look at should have multiple isotopes visible to us, right? Because most of these things are fairly big, so they're going to have some reasonable probability that there will be multiple variants. A monoisotope that's all carbon-12, an M+1 peak that has one extra neutron in it, and so on. So one of the things that we want to be able to do is to group together these ions to reflect the fact that they all belong together. In proteomics, I mentioned that you might see the same peptide in both charged +2 and charged +3 format. You may see it in oxidized form and non-oxidized form. The same thing can happen in metabolites. Maybe not always different charges, because frequently metabolites are living in this +1 world where you don't really get 2 protons carried on a metabolite. It differs by molecular class. But that means that we're going to need to group together different isotopes that belong together, different chemical variants of it as well. The camera software is part of XCMS Online. So this is one of the signs of success for a bioinformatics package is that people extend upon it, that they build new capacities for it. And in this case, we see that camera is able to recognize related chemical compounds that all relate to this core structure. So this annotated peak table is the output from it. You can read that to see what associations it has made. Maybe it thinks that this is the silver adduct and this is the lipid without the silver adduct, and it recognizes these as the same chemical compound. All right. Now, why do we need to bother going further than this? You know, if we can look in an individual LCMS experiment and say these ions are the ones that we care about, we're done, right? Generally speaking, we like to do comparisons. We like to be able to say these are examples of 5 replicates of this cohort, and these are 5 replicates of that cohort. So what differentiates these 2 cohorts? In the example this morning, I was going to compare a triplicate of Coke samples to a triplicate of Pepsi samples. So we would like to be able to compare a particular analyte across experiments. And that requires that we have the ability to deal with liquid chromatographic variability. As I mentioned, there's some amount of retention time drift to say that where this metabolite shows up in the course of the experiment is going to change very slightly. At first, the software uses this coarse matching to sort of say these are big landmarks on the plane of these experiments, and we can say that the big peak over here is the same as the big peak over there is the same as the big peak over there. This mapping among different samples produces a coarse match. But then we need to do more retention time correction, and this process is a fairly complex one. So imagine that you tell the software your mass accuracy is very, very good. It's within 10 parts per million. But in reality, it's about 20 parts per million. Now you have the problem that a particular feature, which could have been successfully matched across runs, no longer matches because the mass of one is too far outside what was measured elsewhere. So setting too tight a mass to charge tolerance may prevent you from recognizing the same feature in another file just because the measurement was slightly askew. Setting it too wide, though, creates the alternative problem. You know, if you put up a sign in your front yard that says, "Lost dog, reward $1,000," you're going to get some people showing up with dogs saying, "Is this your dog?" And maybe it's their dog, but they would really like you to take that dog and give them $1,000, right? So setting your search parameters too broad is also a problem. You don't want to match an ion in this experiment that is not the same as the ion over here. So setting these thresholds really matters. Okay, so when we have compounds that are crowding a particular region of the experiment, a particular retention time and mass to charge value, we can end up with a lot of problems. What if, for example, you have one really big peak that is hiding over in its shoulder a very small peak, right? We've mentioned this problem of major ions kind of masking the presence of lesser ions. Okay, so this is the set of problems that can vex us in this process. Now, retention time alignment is complex. You remember when I sort of stepped back and said De Broglie graphs were complex and I wasn't going to be able to describe them in close detail, but I wanted to give you the general gist? I'm going to do the same with retention time alignment. Conceptually, it's another one of these dynamic programming problems that we met with when we talked about Smith-Waterman and Needleman-Wunsch alignments. So from the initial grouping, the algorithm typically identifies hundreds of well-behaved peak groups in which very few samples have no peaks assigned and very few samples have more than one peak assigned. Such well-behaved groups have a high probability of being properly matched and can be used as temporary standards. They're going to act as sort of landmarks that we use to lay out the scope of the problem we need to do to adjust for this matching. Because the well-behaved peak groups are generally distributed evenly over the significant portions of the chromatographic profile, a detailed nonlinear retention time deviation contour can be built for each sample. Well, now we're getting into some deep squirreliness. So the software is able to recognize. I'm going to start with a pretty basic kind of warping. Imagine that half of your samples ran in 1 hour and the other half of your samples ran in 2 hours. Do we expect that doubling the amount of time required for a separation will change the order in which analytes appear? No. Generally speaking, we expect there's some amount of random variation. You know, maybe two things ran very close this way in one experiment and they reversed that way in the next. But ideally, we're going to expect that simply doubling the instrument runtime does not really change the order of them. However, we expect the peaks to be broader, and we expect the total stretch of these samples to be twice as wide, because we spent twice as much time on the experiment. So if you were to lay these samples side by side with each other, you would see that this one was twice as long an experiment as this one. So the retention times obviously can't be directly mapped. But we could do a linear fit of them and say, if I take half the retention time on my double experiment, it's probably roughly comparable to the observed retention time in my half-time experiment. So that's an example of warping that just involves a linear fit to say the slope on this one is half that of the other experiment. Dynamic time warping, though, allows us to adjust for very small errors in these alignments. Doing a simple linear fit is great when the instrument is behaving very, very well. But when you have an experiment that's behaving more normally, not beautifully, you frequently find that there are little small variations. So this retention time alignment is able to deal with those problems and get rid of the variation to as great an extent as possible. I do not expect to ask you how retention time alignment works in this, but I do expect you to know that it exists and that it's something that is one of the main reasons people would use XCMS. Okay, the next thing that XCMS can do for you is your statistics. XCMS has really good visualization capabilities and test design capabilities. So you can see that Welch t-tests, paired t-tests, one-way ANOVAs with post hoc assessment of p-values, and repeated measures ANOVA are all possible within the parametric set of operations. This means that if you have samples for apples, oranges, and tangerines, and you want to evaluate intensities of particular metabolites that appear in all three of those, something like a one-way ANOVA can deal with that quite nicely. You can also do non-parametric testing. Mann-Whitney, we've called that the Wilcoxon or U-test as well. The Wilcoxon signed rank test is related to this paired t-test. And if where ANOVA assumes that the data are normally distributed. Kruskal-Wallis is much less caring about what the extent to which your data are normally distributed. I don't know the Friedman test very well, but they use that as a non-parametric variant on the repeated measures ANOVA. And lots of visualization tools. So we're going to look at a couple of those. One of them is this plot. So the first is that we have a total ion chromatogram. Now, I've already told you chromatograms are a very, very important concept for today. The total ion chromatogram is quite different. Instead of showing the intensity trace for one particular ion, we're showing the intensity trace for all ions to say how much intensity was seen in these MS scans as a total. And as you can see, we have this kind of blast of noise at the start of these, and then we get some spikes in here. These are kind of where the bulk of the ions we really want to analyze show up. In this case, we're able to show one experiment on the top, one experiment on the bottom, and this allows us to make an assessment of whether these experiments crested in the same places. We also have these values, though. So we have upregulated and downregulated. Those are the two colors we're showing, where green and red are the colors corresponding to that. And they're using the shades and the brightness and the circle radius to tell us about the p-values that come from that correspond to those metabolites and the fold change. So you probably remember a plot when we were talking about microarrays that showed p-values and fold changes on the same plot. Does everyone remember the volcano? So this is an alternative visualization where the circle size and the circle darkness are used to relate these two categories of information. I'm trying to remember what these are called. Was it a cloud plot? I think they called that a cloud plot within their site. Okay. And here's just an example of what happens when you pin down one of these particular circles that comes out of it. Here we see that they've got an ANOVA that is evaluating 5 different strings that have all been put together in one experiment, and we can see how much peak area was seen for glutamic acid across these experiments. So that's pretty powerful. Oh, I did include one slide for flux balance analysis. That's good. So here we are looking at the goal of using a system of linear equations to evaluate metabolic pathways. So this is really taking us into an altogether different kettle of fish. Before, we were looking at these unbiased experiments, discovery experiments in metabolomics, asking which ions are most differentiating between these two. We're going to still return to that problem to talk about how we go about identifying a differential metabolite. But for a moment, I want you to think about the logical extension of a metabolomics analysis. In this case, we have many different metabolites that are all associated with one common pathway. And because it's a very well-studied pathway, we're able to say, well, gosh, metabolite A is consumed to produce metabolite B. Metabolite C is consumed to produce metabolite D. Both metabolites B and D are consumed to produce metabolite E. So if you start thinking of this in a mathematical way, you can derive equations, a system of equations, that describe the interchanges among all of these different metabolites. And flux balance analysis allows us to see how the math works out. So having quantified a particular set of metabolites, we now want to see what systems we can model out of that. Some of the people who are most engaged in this research are people who are invested in bioreactors. So if you are trying to maximize, let's say you're running an insulin factory, right? You want to have as much insulin flowing from the cell cultures you're working with as possible. But under some circumstances, the cells that you've engineered insulin into are putting that energy into cell growth, like they're trying to duplicate more and more copies of themselves, rather than producing a whole bunch of the protein that you want to manufacture. So folks like that really care about flux balance analysis because they want to find the ways to steer the cell metabolism towards producing insulin, not towards making more copies of themselves. Okay, so flux balance analysis, there's a good paper about it right here in 2010. These are These are people who are looking at equations as a way to quantitatively model the operation of a cell. Okay, this brings us to Metlin. This is the problem of how we can identify a metabolite. The primary thing that I want you to know about Metlin is that it is an academia corporate partnership. These companies have given chemicals to the Susiak lab at Scripps in order to build the libraries of spectra of known compounds. And you can see that even in 2014, there were more than 240,000 metabolites for which they had accurate retention times and mass to charge values. There were more than 12,000 that also had MSMS data to tell us what fragments these particular metabolites produce. So that's quite a lot, which means that if you have an ion that you've produced a tandem mass spectrum for, you can search it in Metlin and see if the identity of that compound is known. Very powerful. But where did all the spectra come from? So I've already mentioned all this business, but I would note one important point in the second bullet. Metabolites tend to change what fragments they reveal depending on how you beat them up. If you put them through beam-type collision-induced dissociation at a high energy, they'll produce a different set of fragments than you do if you just sort of tickle them lightly. So, being able to recognize how the fragments differ as a function of the energy you dump into them matters. You don't have to download their library. In fact, you can't. But if you have a tandem mass spectrum, you can send it in via their website and get a putative identity back. So at a moment like this, I find that caffeine is very, very helpful. And helpfully, they have included caffeine in their library. In fact, they have 4 different product numbers to tell you where you can purchase it. Remember that I said this was an academic corporate partnership. So when you do a search in the Metlin database and you get back a compound, if these companies sell it, you're given a direct link to their website to do the purchase. So this is sort of the payback, right? When you engage in corporate research as an academic institution, there's going to be expected to be some degree of reciprocity. They don't just give you something, they give you something so that you can feature their product, and this is part of that relationship that's come from here. Sorry, I'm sort of splashing myself here. Okay, it's too much caffeine, I'll tell you what. Okay, so it's important to understand that these libraries of spectra must be compared in some way to the tandem mass spectra that we collect experimentally. And one of the most common ways in which we do that is by dot products. So if you have an example spectrum and you have your observed spectrum, one from the library, in other words, and one from your own instrument, you want to see, do the ions line up or not? One of the most popular ways to do this is the cosine angle or dot product route. So I would ask you to think of a spectrum as an address. So if you have only 2 peaks in the mass spectrum, 2 m over z values and 2 intensities for it, you could imagine then that we have a intensity for that first m over z value on the y-axis and the intensity for the second ion on the x-axis. So we can draw a vector from the origin 0, 0 to the location that represents the intensities for those 2 m over z values. We can think of this as a vector. If we have a second molecule that we've measured at those same 2 m over z values, it too can be represented as a vector from the origin. If those 2 vectors line up very, very close together, there's a very small angle between them, and thus the cosine should be pretty close to 1 for that angle. If they point very differently, that would in effect mean that m over z1 is really huge in this spectrum, but the second one is very small. And in this one, the second m over z is very intense, but the first m over z is very small. That's a very wide angle, you see. It's a 90-degree angle, in fact. And the cosine there is nearly 0. So that's a case where we have just 2 m over z values. But now I'm going to ask you to take the conceptual leap of saying we actually are measuring 100 or hundreds of m over z values in these metabolites. So now, instead of this nice little two-dimensional space that we had, we now have an n-dimensional space. Maybe it's hundreds of dimensions. But I'll tell you something that's kind of magical about this. Vectors in this 100-dimensional space you can compute the angle between them in exactly the same way. So again, we ask, is the angle between a pair of spectra where each m over z value is a different dimension and the intensity for that m over z is the distance in that dimension? And if the angle is near 0, we say these spectra are quite similar. If the angle is very large, we say these spectra are quite dissimilar. That's one of the most common routes used for asking whether a spectral library entry is the same as our experimental spectrum. All right. They are certainly, while Metlin has produced one of the largest collections, I'm sorry, I want to make this also really clear. Metlin and XCMS are run by the same lab, Suziak Lab at Scripps. So if you're using their XCMS software for differentiation of experiments, you can also use their Metlin database to ask what are the identities of these ions. The Mass Bank of Japan, though, has also put quite a lot of effort into building libraries of metabolic compounds. So it's distributed. They've got 28 different research groups that each operate a part of this database. So you can think of this as kind of a federated data system. That's actually quite hard to engineer well, but it's something that they've done here. Let me see. They've now got about 11,000 compounds, which, as you probably recall, was about the same number as Metlin had accumulated at the one site. And they also collate spectra. Mass Bank is not really about paying for new data generation. They just want to be the site you go to in order to make sense of your data. So they're trying to collate data from as many researchers as they can. National Institute of Standards and Technologies in the United States is also responsible for generating one of the largest spectral libraries. I almost took a job with them, frankly, on a couple different occasions. Some of the nicest guys you'd ever meet in the field of mass spec informatics. So at their last test, they had 121,586 spectra for just under 7,000 compounds. They had precursor ions measured for quite a few of them. Here you can see the numbers of tandem mass spectra that they generated as well. They generally work in positive ion spectra, but it's worth noting, although I spend all my time thinking about protons on ions, that you can also strip them away. You can also produce negatively charged ions. So they measure in both polarities. Of course, lipids matter, but I'm thinking our time is getting a little short, so I'm going to punch ahead to the finish line on this one, I think. I wanted to note that spectral libraries are not the only way that we identify tandem mass spectra. I mean, libraries are great, and if you have a library spectrum to compare against, that's really one of the best scenarios for you to be able to determine what metabolite is this. But there are also ways that you can apply all the lessons we've learned from proteomics to this problem of identifying lipids. So I really want you guys to know that you don't have to have a library spectrum to identify a metabolite. So in the same vein, my research group at Vanderbilt, one of the last papers we published before I came here actually, was about the Greasy software. It's intended to be about lipids, so it's grease, but it's easy, right? Greasy. I thought it was hilariously funny. The grad student didn't like it as much. That's okay. He had another idea for the software, but I always thought it would not be quite the right name. It's all right. So we wanted to be able to say, for this set of parameters, what are the lipids that we can envision, right? We know that phospholipids have this relatively small set of head groups, PC, PE, whatever, phosphatidic acid, whatever. And we would like to be able to extrapolate from this set of allowable fatty acid chain lengths and head groups and ways to connect them, that we can extrapolate all the lipids that would meet this description. Then we want to predict what the tandem mass spectra should look like if it were this lipid. You remember we did this for peptides, not just yesterday actually. For a given peptide sequence, we wanted to model what fragments should we see. In the same way, we wanted to say, for this set of lipids, what should the spectra look like? So from there, we can do the same thing that we do in database search. We say, which lipids are close enough to the observed mass of this lipid for comparison. We can then project what their spectra should look like and compare them to each other. So the way that you do this evaluation requires a few tricks, right? There are different bond types that connect a fatty acid to the backbone. There's a saturation rule, because sometimes you care about saturated fatty acids only, and sometimes you want to deal with unsaturated as well. There's a range of lengths for fatty acids. We need to then figure out how many can be connected to a particular backbone. So cardiolipins can attach to 4 different fatty acids, while something like a phosphosphatidylipid can only connect to 1 fatty acid at a time. So we want to be able to construct, by attaching the right head groups to these lipids, to be able to construct them. So this is the broad search space that is defined by the user, and from there we can say these are the sets of lipids in the search space, what are their masses, and what should their fragment ions look like. And then we can compare all the observed spectra to those that we've modeled. So you can see that when we took a look at how we do with known spectral libraries, we were able to recover the correct identity an awfully large fraction of the time. So this was very, very promising for this kind of comparison. In any case, I want you to keep in mind that you don't have to have a spectral library in order to identify tandem mass spectra of metabolites. You can use fragmentation rules that have been derived from looking at lots and lots of such spectra before, and that way you predict what the spectra should look like for these compounds that you can envision. It really only works well, though, when you have a set of compounds that's pretty well constrained, a set that have been studied well enough that you know what kinds of fragments they should produce. Okay, so mass spectrometry intensities can be variable, but they still yield peak areas in these chromatograms to quantify peptides, lipids, and metabolites. You can use them in all 3 of these cases. Identification is quite a lot harder when you step outside of the classes of molecules that have sequences, and that is frustrating, but it is not insurmountable. Spectral libraries have played a key role in recognizing metabolites, but I think the further along we get in the world of metabolomics, the better we're going to get at being able to model what fragments we should see when a metabolite blows up. All right. That brings us to the conclusion of quantification in proteomics and metabolomics. Tomorrow morning we are back upstairs. We're back in the seminar room, and we will be discussing image processing and flow cytometry. It should be very interesting. And then after that we have just one more day. So I look forward to seeing you all tomorrow morning, bright and early, 9:30.